My name is Nathan Tucker. I'm an assistant professor at the Masonic Medical Research Institute in Utica, New York, and a visiting scientist at the Broad Institute of Harvard and MIT. Thanks to the organizers of this wonderful seminar series. I've enjoyed every single one I've had the opportunity to catch, and look forward to catching up on a lot of the others on the YouTube channel. I originally gave this talk back in on June 12th, I believe, on a Friday, and had significant audio issues. Hopefully they're mostly resolved. I'm retaping this introduction to forewarn some people. There's a little bit of microphone noise going forward. I did my best in video editing to remove some of that, but at least from a baseline of being able to hear my voice so I can actually explain my figures to everyone, hopefully this is much superior. So the following is going to be explanation of data from our group that was recently published in Circulation, dealing with single nucleus RNA sequencing from non-failing human hearts. I'll have a one-slide addendum on the end of another research letter that was just published online in Circulation, I believe this week of June 22nd, that where we tried to delve into some of the cardiac-related phenotypes associated with coronavirus infection and see what information our single nucleus RNA sequencing could provide in relation to that. Two goals. One, I hope to walk through our data, explain what some of these figures mean, talk a little bit about how all of these libraries are generated and the like, the decisions we made and why. And then number two, hopefully, inform people so you can interpret these data in a proper way. We didn't want to lead people down the wrong track by our analyses or overstate any of our claims. So on the whole, we tried to be very conservative about conclusions we were going to make. But in the following section that I'll splice in here in a minute, I'll try to explain, some of the caveats and pitfalls and things that we had to do to work on the data set and provide the data that hopefully you all find is a wonderful resource. So without any further ado, I'll end my introduction here and start right into the presentation. Thank you. The past decade or so, RNA-seq has been a wonderful tool to understand some of the molecular mechanisms underlying a given phenotype. So I have two examples here. One's actually a microarray study technology that predated RNA-seq by a little bit. And here are the authors from University of Pennsylvania, good collaborators of ours, took left ventricular samples from dilated cardiomyopathy. They're circled in the red region. It's non-failing left ventricular samples in the blue, and then plot the global transcriptional representation in the PCA plot. See on PC1, the non-failings and PCMs clearly separate from each other, illustrating there's difference between them. You can also look at the gene levels. My study released last year on HFpEF versus controls, and you'll see what particular genes might be up or down when you compare your disease condition to control. So it's been wonderful and hugely informative for a lot of stuff going forward. One small, I guess, significant weakness that we and others have tried to address in our study and others as well. So the problem is these analyses are all done at the tissue level, and tissues are complex. So look at this path image. Here's the left ventricle. There are a couple of infarct regions. Where you pull your sample from will have a huge impact on what the transcriptome will be for your RNA-seq experiment. So if I were going to sample this largely unaffected wall versus something right within infarct, and compare them, transcriptionally would be very different. And then if you want to compare to an outgroup like a non-failing ventricle, where you pull would certainly color your view of what you think the difference is between someone with ischemic disease here versus a control. So you want to break it down to two things. One would be, are a given cell type different? Is a cardiomyocyte here than one in the infar? Or is the cell representation different between those two regions? And by bulk RNA-seq, it's just not really possible to do. Zoom in a little more in this histology slide. Vast majority of this pink is muscle fibers, but in between each one of those, we know there's a bunch of fibroblasts, resident macrophages. You can see some adiposity here, vascular tissue. So they're complex, but they're not uniformly complex. So where you sample is important. So how can we deal with some of these issues to help our understanding what's going on? So our goal, which I guess based on my belief that good scientists, at least good bench scientists, are also good cooks or bakers in this case. So you have your batch of cookie dough or cookies. You want to understand what's different between two batches. You go back to your components and you see what particular component might have made that difference. In the end product, our less appetizing example is to take the 4 chambers of the human heart and break it down into the various cell types that comprise it. So how do we do it? A few choices. First, we chose to do single nuclear RNA sequencing. Some of the pioneering work done by Roger Flu's lab, which is where this figure actually comes from. So reason to use single nuke rather than single cell. So a couple big reasons. Single nuke, nuclei will survive freezing in long-term storage, where cell membranes won't remain intact. So we're able to use tissue that's been collected over the past 10 or 15 years to run through these assays, as long as they have good RNA quality. Cell shape is an issue. So when we're using microfluidic platforms, if we have a channel size that any cell or nucleus needs to pass through. Pretty much are huge. So just based upon what will pass through your device, you're selecting for against given cell types. And we didn't want to do that either. Another important component, I guess, to think of this as nuke-seq is largely sampling nascent transcription. So what the cell is trying to make from a transcriptional standpoint at that time. Plus and minus, because what you're not seeing is anything with the buffering or RNA half-life that occurs in the cytoplasm. But you do get a good picture of what the cell wants to do at that moment. Yeah, so those are the big reasons we went with nuke, and then we had to pick an approach. So the original approach was to do basically a single cell sort into a well, do the library prep in isolation, really deep information out of each cell, but extraordinarily expensive to do these protocols. If a microfluidic based approach, this was the original drop-seq, where you barcode each cell's transcriptome. The fluidigm approach, it's also 2. And then more recently, some spatial approaches that take some of the labeling approaches used in these other highly multiplexed efforts, pattern them over top of the slide, and then try to do spatial registration of where a given transcript came from. We're really excited to see where this goes in the next couple of years. So we choose a microfluidic approach and chose the 10x genomics-based system. This is a big study. And as such, we wanted to introduce as little of variability in that library generation step as possible. So while we did some homemade drop-seq style stuff using the 10x genomic system, really was helpful for getting good reproducibility of our data from batch to batch. So this is what the machine looks like. Inside the tray here is 8 devices that you can run at a time. The runtime is 7.5, 8 minutes. Pretty fast. Looks like a toaster, but it's very unimpressive when you see it on a bench. But if you're going to zoom in on these devices, it looks pretty cool. So basically what you're seeing is the dark dot, or the larger dark dot here is a bead that contains barcoded oligos that capture and label any transcript that it's going to see to say, hey, it came from this bead or this droplet. And then down the side, hopefully you can see a small little cell that will get actually in the same droplet as that bead as it comes down through. That's your ideal scenario. So cell goes into the droplet, sliced, releases all of its RNA. All that RNA is captured and then labeled by the special barcode on the bead. After you do library prep in aggregate, you're able to guess, deconvolute the aggregated signal back into what came from what individual cell. So this is from the original dropseq paper. I suggest people check it out. It's really a landmark paper and really well documented for what they did. All right, so let's get into the data about the 4-chamber map. We had about 290,000 cells from the 4 chambers of the heart. 7 individuals, 4 women, 3 men, all the chambers. There's one technical variation in one of the replicates that increased the number of transcripts that we were able to get from a library preparation. It's in the methods of the paper. I suggest including it for people doing NUTC. You go through quality control workflow. So you take all your samples, you push it through standard pipeline, and then we drop at the sample level. You know, these preps from human tissue samples are challenging. We do lose a significant portion of them to quality control, especially atrial tend to be very difficult. Pass it through cell bender, I'll talk in a minute. Filter and aggregate at the cell level after anything that passes through the sample level. So if you see cells that are acting oddly, we get rid of them. And then do some kind of batch alignment I'll talk about also. So CellBender was a tool that was developed by Natash Babadi's group in the data science platform of the Broad Institute, primarily by Stephen Fleming, pictured here on the right. And so what it does is, usually whenever you generate an experiment, you have a UMI barcode plot like this. So UMI is the number of unique transcripts, and the barcode is just basically the cell as you pass down through here. So each one of these cells has a number of transcripts that goes with it. And normally what they look like in a whole cell experiment is you'll see a large plateau of everything that cells and are very complex, drops off a cliff, and then you'll see a little plateau down here, and that's just ambient RNA floating around to your supername. And nuclei experiments get a little trickier because you're going to think you're lysing 90% of the RNA from a cell out into the supernate, and there's a trade-off between how much you want to rinse versus, you know, the integrity of your nuclei or how sticky they end up getting the more they handle. So instead of these sharp drop-offs, in this case, you're going to see kind of a little, I guess, shoulder here that would suggest dropping off. So one of the first protocols, you say you put a hard line right at that shoulder, and you say everything above is a cell and everything below is background. But what Stephen Mirtash did is said, actually, let's look at each one of these cells, see how different it is from the ambient RNA profile, and if it's sufficiently different, we'll call it a cell. So if we were just going to take the most complex things, you'd still get your cardiomyocytes and fibroblasts and the like. That's the orange and blue lines that you see here. But we start to pick up, you know, some of these things down here that are less complex, you know, or anything, you know, down below 1,000 unique transcripts. We would have thrown them away by any approach other than this. So by this, we're able to pick up much more information and save a lot of these expensively generated data. The other thing CellBender does, it tries to quantify the background and actually remove it from each cell. You know, there's 100 or a couple hundred transcripts in each one of these droplets. It's just going to be background. So Mirtash and Steve's approach attempts to remove that. We do batch correction. So this plot, each one of the dots on here is colored by individual, and you can probably appreciate that the individuals tend to separate really well. That's problematic when you want to do clustering and say, let's look at cardiomyocytes in aggregate across individuals. It's tough to group them together when the data look like this, even if it makes for a really beautiful plot. So we use a tool called SCVI that attempts to align those individuals. Basically what happens is, you can appreciate the colors are starting to mix in a lot of these large clusters much better than was done previously. It is absolutely a necessary step for working with human data. A lot of times with mouse, rat, other model system data, you can get away without a batch correction, like at least SCVI-based batch correction. You can get away with a little more crude approaches, but for human data, we found this to be absolutely necessary. Finally, we want to identify non-nuclear cells. This seems to be a quirk of cardiac work. So, given there's nuclei, a lot of our reads end up mapping to intron. So average percentage of exonic mapping reads is about 10%. But if we see clusters or cells that deviate from that significantly, have many more exonic reads, we think it's indicative of cytoplasmic fragments or contaminants that end up passing through our library generation step. You have to think there's a bit of sarcomeric apparatus that's still floating around even after we filter, and that'll be treated as a cell, as RNA on it. So, but at least at this stage, we're able to remove them, and then we're just comparing nuclear transcriptomes of cardiomyocytes versus cardiomyocytes instead of adding the noise of cytoplasmic RNA. So this is what the map looks like. It's called a UMAP plot. Each one of the dots that are kind of fuzzy here, there's so many, represents a cell. The distance between any two dots in this map represents similarity. And then the color on here would represent a cluster or a cell type that we would then assign later. So we look at the, perhaps the things that mark a given cluster as a marker gene, and then we can assign all these labels for cell types to them. And I think what we end up learning, no surprise for the types of cells that we saw, but the amount of information that we learn from each cell subtype, what makes them unique is pretty astounding. So most of the analytic work I'm going to talk about after this point was really heavy lifting was done by Mark Chaffin, who's one of if not the most talented analyst and statistician that I've had the pleasure to work with at the Broad Institute. So with my more biologically inclined background and Mark's more computational analytically inclined background, I think it made a great team for a study like this to come together. It's a good point to take a pause about what you can or cannot say about these data. So point one, if you want to look at one gene and compare it to itself relatively across different cell types, totally fine. But what you can't do is compare the levels of two genes to each other. So unfortunately, the amplitude that you capture for gene expression isn't on par with what you think for a TPM for a traditional RNA-seq experiment. There's some bias that's introduced in the library prep. So we're doing poly-DT-based capture. It'll capture the polyadenylation signal, but it'll also capture homopolymers within the introns of the transcript, especially. So you can imagine the amount or length of those homopolymers will result in a different probability of capture for a given transcript. Reverse transcription, long transcripts, especially with the nuclear transcriptomes we're dealing with, or transcriptomes with odd runs of bases that the RT enzyme would have trouble with. You'd get basically not a linear transformation of abundance to actual readout. So you can't compare different genes to each other. When we're doing our differential expression testing, we generally, just for conceptual purposes here, most of the information is at that cluster level. So each individual cell is only going to have 1,000, maybe 1,500 transcripts associated with it. But when you collapse, let's say, nearly 100,000 cardiomyocytes that we pulled out of this map down into an entity, the depth of understanding is on par, if not better, than what you'd get out of a traditional RNA-seq experiment. So when you're comparing things, for the most part, I guess in concept anyway, you're comparing at the cluster level, not from an individual cell. Totally able to look at low abundance transcripts. You just need to be sure that you have a sufficient number of cells that you're looking at. So it's a probability event. If you're looking for a lowly expressed transcript in a low abundance cell, it's going to be a lot of work for you. But for example, you want to look at something that's very lowly expressed in cardiomyocytes, it should be totally fine. We don't have isoform information. We are doing nuke-seq, so introns are mostly retained, but even traditional cell capture, that's 3' capture, so you get a read pile up about 500 bases upstream from that polyadenylation signal. There are some non-standard library prep protocols being, in process or in development by PacBio, Oxford Nanopore to do long read sequencing and start to pick up isoform information also. Subclustering, so beware, especially if you start to see these big clusters with a lot of cells in there, like our cardiomyocytes in this case. The way the clustering algorithm, it wants to break those apart. if there's a lot of cells there. And we're not confident that that speaks to biological variation as much as kind of the nature of the algorithm itself. So if you do see clusters that break apart like those cardiomyocytes, and you believe it's true, go validate it. You need to use an orthogonal approach. Whether it's in situ is our standard method to do this, but another sequencing-based approach would be appropriate as well. Point two, if you see nuclei that exhibit properties of other cell types, particularly cardiomyocytes, be wary. There's contamination. We use CellBender, but you don't want to overfit the data. So there's a little bit of dirt, I guess you could say, that's left in on purpose, but that can make it look like other cell types, have signatures of others. So if you see myocyte genes in macrophages, then you believe it to be true. Again, go validate it by an orthogonal method, please. Our goal here is not to send some poor graduate student or postdoc on a wild goose chase, chasing down an artifact of the way that we generated these data. So we think it's an outstanding approach for discovery of cell types, transcriptional profiles, especially relative to other cell types for them. But it's not made for absolute quantification. So I think there's a lot of worthwhile data here, but if you want an absolute number, you got to use a different approach. Let's get into definitions of cell types, what these data look like. So we'll take atrial cardiomyocytes, these orange ones here as an example. You can look at these dot plots. The larger and darker the dot is, the more highly expressed it is. So if we look at some of our favorite genes, MPPA or white gene 7, heavy gene 6, see a dark dot in the atrial cardiomyocyte column. This speaks a little bit to that contamination thing I just talked about. So I think we largely appreciate the AMPs produced by cardiomyocytes, but you're seeing dots in these other cell types. If you think it's true that intrinsic cardiac neurons are producing AMP as well, go validate it. It might be true, but it also might be contamination from just the other atrial cardiomyocytes releasing their cytoplasmic components into solution. These are top 6 genes. There's many more that mark specific or sensitive markers of each population. You can plug them through ontology. I don't find the terms particularly interesting for the most part, but they can help with cell assignment or label assignment if you want to use it for that. So you can do this for all the cell types that we see. You get pretty good markers for all of them. The best way, this is a visual representation, but the best way to actually look at the data is in tabular format. So I encourage everybody to go to the supplemental tables of our manuscript, and it'll have everything you could ever want about what a marker is for a given cell type. I just warned you about subclustering, but there are examples we feel good about. So we took the endothelial cells and pulled them basically out of all of the others and reclustered them in isolation and found 5 subclusters colored on this map here. This one marked X, has that shift of more exonocrines than we would expect, so we removed that. It's also in a weird space on the UMAP plot. But I end up with these 4 other populations. EC1 and EC2 we think are probably microvascular associated endothelial cells. EC3 we think have hints of endocardium, but we didn't validate in situ, so don't hold me to it if I'm incorrect there. And then the 4th I was pretty excited about, looked to be lymphatic endothelial cells. I think the lymphatics are a bit underappreciated in the cardiac context. So, I think through this, I think we provide, probably the best transcriptional definition of lymphatic endothelium in the heart to date, and we hope it can serve as a tool for other people who are going to do downstream work in that area as well. It's just defining the heart. Now let's start comparing things. So take that UMAP from all 4 chambers, break it into the respective chambers, and start to do cross comparison. So what's different between the atrium and the ventricle, for example? So we can take each one of these dots is colored by the cell type from which the comparison was performed. So orange myocytes, blue fibroblasts, red are paracytes, pink are macrophages, and I believe purple or endothelial cells. They can display like this in each individual gene. You know, more left, it's more atrial, more right, it's more ventricular. But you know, I have trouble pulling information out of a plot like this, other than saying a lot of things are significant. So we thought of an alternative visualization approach for these data. It comes out with these dot plot style graphics here. So let me just run through an example to help illustrate how they work. So you take your gene from any of these columns here, and in it you're going to have the 6 potential comparisons that we considered for this. So the color will represent the direction of this effect. So if it's more red, it's more towards the term on the left. If it's more blue, it's more towards the term on the right. Size of the box will indicate the level of significance for that given comparison. So if we take A and B, it's strongly red, so it's strongly up in the atrium. Sidedness doesn't really matter. It's up in the LA and RA also. So compare left to right, there doesn't seem to be a lot of anything there. If you see a gray box, that means we didn't have sufficient number of reads to actually do the comparison. There's so little AMP in the ventricles, it's just impossible to test. The other thing, if you see a dot in the middle, that means it reached our multiple testing threshold. So significant differential expression in that case. You can have A2, which is the opposite in cardiomyocytes. So blue, so it's high in the ventricle. Same both LV and RV compared to the atrial compartments, and no laterality here either. But then, you know, these are standard ones, but then you see examples that are really interesting. So AMP is an iron homeostasis gene. The A versus V comparison. It's up in the atrium, it's significant. But you know, LA versus LV, it's gray. There doesn't seem to be a lot on that side to even compare. RA versus RV, strongly up in the RA, and LA versus RA, it's blue, so it's high in the RA. So I think what AMP ends up being is a pretty specific marker of right atrial cardiomyocytes. So by looking at these plots, you're able to pick up kind of unique patterns. You know, here's AMP on it. I have my favorite PIDX2 that I've worked on. It's next to the top association locus for AF. And in this case, strongly up in LA versus LB, strongly up in LA versus RA. So it seems to be a left atrial specific cardiomyocyte marker as well. So I encourage people to just sit back and look and try to identify patterns like this. But the full data, again, are found in the supplement. Cardiomyocytes are different between chambers, which I don't think is surprising to anyone, but we wanted to look at other cell types. So we tested fibroblasts and endothelial cells and our macrophages, stripped the gene labels off so it didn't distract us. But again, it's all in the supplemental table. So you just step back and look at the number of dots here for all these comparisons. I think it's striking the number of things that are chamber or side or A versus V specific when you look in these different cell types. This is not an exhaustive list of everything. So really underappreciated diversity in transcriptional profiles of these support cell types as well. And we're curious what that'll end up meaning for physiology going forward. Finally, let's try to use these to inform some disease biology. So we took 2 approaches. Take your large map, use information we have from population genetics, and do a technique called LV score regression. What that's going to do is examine the heritability, you know, estimates for whatever GWAS study, and say nearby to loci are there genes that happen to be specific for a given cell type. So another way to think of it is, are there cell types or specific expression in cell types that seem to drive the heritability of a trait? So an example of QT interval, you know, dealing with ventricular excitation through repolarization, that seems to be driven, in this case, in this plot, by cardiomyocyte-specific expression. So the rule for QT interval is that cardiomyocytes drive it. In these plots, the blue dotted line is just the 0.05. The red line is going to be your multiple testing adjusted threshold. So you can do this for anything, basically, that has GWAS, available GWAS summary statistics. So you can go on the arrhythmia side, you have the QT interval, we have the PR interval. So you can atrial conduction, in this case, to the nodes. So again, you have a strong signal for the cardiomyocytes, but you have these subthreshold ones, things like adipocytes, that are now well recognized to be signaling mediators, vascular smooth muscle cells, what we think are intrinsic neurons. So it's a little messier control of PR interval as compared to QT interval. And then you get the complex traits or diseases like atrial fibrillation. And, there's nothing that reaches significance thresholds. Strongest signals in cardiomyocytes. But I think what this speaks to is the complex mechanisms of getting to that AF phenotype. So you can imagine if you had part of it that was governed by the PR interval, another mechanistic component that operates through an entirely different mechanism, that'll do is pull your hypothesis, in this case, towards the null. So it drowns out a little bit of any specific signal that might be due to like an endophenotype such as this. So what you want to do with complex trait like this is either help stratify your GWAS by mechanism if at all possible, or look at these endophenotypes instead. Applied to other traits, CAD, you would think vascular associated expression, and we do see parasites and endothelial cells being the top markers. LDL governed by the liver. We do see a signal in adipocyte specific expression that relates to LDL. The negative control to diabetes has impact on cardiac function, but it's not expected to be driven by any expression in cardiac cell types, and we don't see a lot of signal there either. Alternative approaches to take things we've learned from Mendelian inheritance, and those tend to be the things that are on clinical testing panels, and say, hey, where are these genes of known importance expressed? Are there any interesting findings? So you can just do a dot plot of this is 106 cardiomyopathy genes, and you see probably the majority of them have cardiomyocyte specific signatures, but there's a few outliers that are worth noting. So here's like ryanodine receptor, strongly expressed in cardiomyocytes, tropomyosin, so in myocytes and vascular smooth muscle cells. Then a couple interesting examples. MAP kinase seems to be strongly specifically expressed in macrophages. So maybe we want to revisit the mechanism that we think this might confer the cardiomyopathy phenotype. Or LAMA4, which is most strongly expressed in adipocytes. Is that the mechanism of action there? So conclusions this part, 290,000 transcriptomes, 9 what we call major cell types, about 15 in these superclusters, and subcluster to your heart's content. We tried to be very, very, very conservative here. So encourage people to go dig at the data if you think there's more there. Marked chamber specificity of expression profiles, and not just in myocytes, but also in these other support network for cardiomyocyte function that has been previously a bit underappreciated. And then, starting to use these data to identify new mechanisms, rules of disease risk inheritance, inform modeling for target discovery. The one thing I think back to my grad school or postdoc days, and you're working on a gene and you're looking for a phenotype, and one of the possibilities is that it's not expressed where you're looking necessarily. So if you use this map or similar maps to say where's my gene of interest actually localized, hopefully it helps with your modeling to help capture any phenotype that may be present. More info. This paper was released online on the 14th of May. So all the supplemental tables will be available there. Apologize for the size of the PDF right now for the main. But we also release these data to a single cell portal of Broad Institute. So all you need is a Gmail account, uses for authentication. You can go into the single cell portal, search heart. You'll find our study, SCP-498. It's another identifier. Within there, for less computationally inclined people, you can just type in your gene of interest and see what cell types it's expressed in, or compared to the others. You can do it in that UMAP overlay format, or what we call violin plot, or box and whisker. You also have the opportunity to download the data through here. It'll be post-QC data. I would suggest that as a start point for people who are going to work with it. The QC for these were a significant endeavor. But yeah, this download button will be blue for you after you authenticate with your Gmail. And then we're also releasing the data on dbGaP and SRA or ANFIL for people who want to go back to the raw sequencing. All right, a quick aside before I wrap up. We wanted to, we're in the midst of revisions on that paper and working on other studies, and coronavirus happened. So three major questions, I think, related to cardiac phenotypes is, one, there's a lot of significant amount of mortality, about 20% associated with cardiac complications. So we want to understand or help to understand, why that might be the case. There's cytokine storm hypotheses. There's primary myocarditis hypotheses. So let's see, can we shed any light on that? Two, there seems to be a greater susceptibility of people with pre-existing cardiac conditions to severe cases. Is there any reason for that we can derive from our data? And then three, some amount of debate about the continued use of antihypertensive, such as ACE inhibitors or angiotensin receptor blockers, and see do they have any effect on known receptor for SARS-CoV-2. Let's start with number one. So in the Preexisting project, we wanted to look at expression of receptor, which is ACE2, and then associated proteases that help prime the virus for uptake. So ACE2 seems to be expressed most strongly in parasites. Makes sense. There's a lot of microvascular damage, both in the lungs and the heart after infection. But there's also appreciable expression in fibroblasts, vascular smooth muscle cells, and various cardiomyocyte populations. TMPRSS2, one of the proteases, we didn't really see appreciable expression, but CTSL is pretty widely expressed across all the different cell types. So speaks a little to the cell types that at least have the possibility to uptake the virus. Next, we wanted to see about the pre-existing conditions. So we were doing a study comparing non-failing left ventricles to those from individuals with dilated or hypertrophic cardiomyopathy. When you look by bulk RNA sequencing, no significant difference in that ACE2 receptor expression at all. I guess these results are a little in contrast with what other groups have shown, but we had over 10 samples from each one of those that were, collected by the wonderful protocols as if for transplantation for DCM and HCM explants by the team at the Magnet Consortium under the guidance of Ken Margulies at the University of Pennsylvania. So we're pretty confident in the quality of these samples. So no difference at the bulk level, but what that bulk thing is hiding is an interesting pattern. So if you look, non-failings in white, DCM in blue, HCM in red, and fibroblasts, and parasites, and vascular smooth muscle cells, this receptor goes down in the presence of cardiomyopathy at a cell subtype level. But the outlier to this is in cardiomyocytes, where you have a pretty striking and significant upregulation. This is log transformed as well, so a large amount of ACE2 expression increase in cardiomyocytes specifically. So if you think about combining all of these together, in what would be that bulk RNA-seq experiment, the down in all these cell types, the most numerous and most highly expressing cells are cardiomyocytes. They end up masking the signal to be this, whereas really it's down, 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 up. So kind of a interesting use case for something that you could really only uncover by really complicated in situ style experiments or an approach such as what we did here. Finally, ACE inhibitors, long-term ACE inhibitor use, expression of that. There was guidance that there should be no change in clinical care for folks on ACE inhibitors from European Society for Cardiology, American Heart Association, American College of Cardiology. So we looked at ACE2 expression on when people with and without ACE inhibitors. Unfortunately, it's only limited to hypertrophic cardiomyopathy cases. It's the only ones where we had sufficient number in each bin. But the important takeaway is that there wasn't a big difference based upon ACE inhibitor use. So I think supportive of the message previously laid out, at least from these molecular data from the cardiology societies. so where's this going? You know, we're continuing on different model systems, and they work on some interesting models that use these type of data outside of the human stuff. There's a lot of orthogonal approaches. If you look at chromatin accessibility, I think we'll be at histone marks relatively soon. The isoform stuff from long read sequencing should be really fun to work with. The spatial things are going to be amazing in the next couple of years for looking at unique anatomical features and, you know, cell-specific expression there. So I think ultimately this is going to end up, you know, obviously it's more expensive than bulk RNA-seq experiment now, but as the cost comes down, I think, you know, it's going to serve as a replacement for some of those figures. So they're complicated experiments, both to perform and to analyze. So I encourage anybody to reach out at any time if you have any questions, want guidance, or formal collaboration on any work related to this. Acknowledgements, you know, let me start with the middle. The work I presented today was performed under the umbrella of Precision Cardiology Lab, headed by Patrick Eleanor and Christian Stegman. The PCL is a joint venture between the Broad Institute and Bayer. So I think it's a testament to academic pharmaceutical collaboration to do a study of the scale that brings the strengths of both sides together to get it done. So I certainly appreciate the target-minded focus that pharma brings with their intention for rapid translation, you know, especially when we start getting into the technical aspects of data generation or analysis. So it's been really fun working with their group. Also, as part of that study, you know, the GTEx headed by their director, Kristen Ardley, Francois Aguet, who developed the tool to look at exon versus intron ratio, was absolutely essential to the study. It helped immensely. University of Pennsylvania, Ken Mergley's, Ken Beatty, supplying all the tissue, wonderful collection, wonderful metadata. And when we start talking about clinical phenotypes or other biological pathways, when we are down in the weeds of the data, Ken and Ken had really wonderful ideas of things we should be looking at, or things that we perhaps took it for granted as, oh yeah, of course it would be there. That way, they'd say, maybe you should think about that a little more. So I really enjoy all of our calls and the collaboration we've had with Penn as well. And then the various funding mechanisms. I also want to acknowledge my nascent lab at the Masonic Medical Research Institute. So I showed up in March, coronavirus hit, and then we transitioned from a research laboratory into a clinical testing laboratory. So at the time of recording this, you know, we're running somewhere between 1,000 and 1,500 clinical tests a week for a local community hospital. So Ryan Pfeiffer and Myri Desai are the people downstairs as I record this, running these tests to help the local community. Really kudos to the team effort, both at the Institute and within my lab, to make sure that happens. We're proud to be able to help, although we hope to be able to get back to research soon. Coming soon, a couple computational biologists that will help support you know, this and other associated low input techniques in the lab, you know, both for stuff that we're doing and hopefully for some collaboration as well. So with that, you know, if anyone wants to follow up, email is ntucker@mmri.edu. You can find me on Twitter, nr_Tucker. And, you know, I'd be glad to discuss more of this with anyone who's interested. So thanks a lot for watching. Apologies for the technical issues with the first recording, and hope to talk to some of you all in the future.
Nathan, can I start with a question? You've got quite a lot of questions that are up in the Q&A box, but I thought, you know, for the benefit of especially the listeners, at the part specifically where your voice was kind of fading back and forth a lot more, was when you had the table with all the squared colored, all the colored squares. Would you mind just going back to that one? Because I thought that's really an important figure, and you know exactly what the representation of the different colors and the different sizes of circles, or even the colors of the different sizes of the colored squares was quite important to understand. Yeah, this one here.
This example here.
Yeah.
Okay, so I'll confirm that my audio is good right now before I start. So basically, this is a different way to represent what would normally be a volcano plot, to break down all of the comparisons that might be present rather than plotting 6 volcano plots. So what we want to do is look across these genes and do all 6 comparisons in this way, and to run through the example quickly. Things that are more red, the boxes that are more red and larger, are more significant and more strongly expressed in the term on the left that's also shaded red. Things that are blue are obviously more strongly expressed in things on the right. Things with a dot reach statistical significance.
Yes.
Example of MPPA, strongly expressed in the atrium, no real difference between left and right. So you can see all these atrial versus ventricular comparisons are strong. Inverse is true for A2, which is strongly expressed in the ventricle, no difference between left and right. And then you get interesting examples such as HAMP2, which globally looks larger in the atrium, but seems to be entirely driven by expression within the right atrium. So strong in the right, weaker in the left. So if you go through plots like this.
What does it mean when there's a gray square in
Sure. So the gray squares, the one issue you have with these data is that the statistical models have trouble in general handling the value 0, and things that are entirely absent we're not able to test. Okay. So in this case, if there's no expression in the ventricle, you're not really able to compare between them. It's almost better in this case that it's a little contaminated, because at least you have something to compare against. So you can apply those globally here.
Fantastic.
Hopefully that makes a little more sense for everyone. Yeah, I'm sorry for the audio.
I mean, it's always so amazing how it's possible to represent that much data on a single figure. It's like a picture tells 1,000 stories kind of thing. I'm not getting my analogies right here. So let's go to the Monica Gladka is asking you or saying wonderful data, thank you. Do you think that single nuclear sequencing will underestimate cellular heterogeneity within the same cell type? Or perhaps it's the opposite, and what is the average gene number per individual nucleus that you've got there?
Okay, let me start with the last part of that first. Average gene number or transcript number that we observe is highly dependent upon the cell type. Cardiomyocytes you can often capture by this methodology upwards of 5 to 10,000 transcripts from a given cell. Whereas you look at immune cells and we're down in the hundreds. So it ranges depending upon the cell type that you're looking for. But on average, around or a little above 1,000, the new chemistry from 10x Genomics actually improved that significantly. So I encourage everyone to use V3 rather than V2 if you have any laying around. Do I think it underestimates cellular heterogeneity within the same cell type? Not necessarily. I think I think that's a really good question, and I think about it a lot. There are 2 concepts that go into this. One is there are 2 distinct cell types that fall under the same window, and I think singular nucleus RNA sequencing is really good at capturing that. And then the second possibility is that within that same cell type there is a continuum within there, and in that case it almost needs its own metric of heterogeneity within that cell type. And we've thought a lot about how to deal with those data or how to display that metric and aren't quite there yet. I think it's possible to treat it as a metric, but again, all these things are very complicated because it deals with your clustering resolution and how you're treating your data before that. So I don't think it inherently underestimates it, and some of it's powered by the number of cells or nuclei that you do, but requires a tailored analytic approach.
And also the choice of using the 3' end of sequencing could affect that too.
Oh, for sure. So I will say too that if you want to use a different methodology, that this is a low complexity that you gather by this. If you want to go back and hammer a given cell type to understand more about it, you can use this as a discovery mechanism and go out with something that can look at full isoform coverage and the like thereafter. And I would encourage people to do so.
Okay. Okay, next question. Says, very nice talk. Is it valid to claim that when cell clusters express higher MPPA than the other cluster, then they are likely atrial cardiomyocytes? And if so, how many fold increase should be used as the cutoff for MPPA, for example, or other genes?
So it's a good question. I think so. The quick answer is when cell clusters express higher MPPA, not necessarily. I will say that the contamination of cell types or nuclear types or clusters is not equal. It's a proportional contamination, and I think that you really have to consider each one of these in isolation when examining whether or not they're atrial cardiomyocytes or not. I mean, I will say when you look at these data, you'll see a level of MPPA in a cluster that will leave no doubt in your mind that they're atrial cardiomyocytes. But the other thing too is you're going to have all these other marker genes that will, definitely tell you that they're also cardiomyocytes. There's no single gene analysis really that goes into the clustering. It's everything. And the other end, we don't use cutoffs. Everything is a continuous variable to us here.
So Nathan, your next one is from Anand Chopra, whom I'm suspecting is someone who works with you with a face on the screen.
Hi, Anand.
Okay, because he's saying here, Monica, I'll let Nate answer as well, but believe we got between 500 to 1,200 genes per nucleus in this data.
Yeah, that's true. I will say our new data is much, much better. So like I said, use V3.
Mm-hmm. So another point, excellent talk. Just wondering when the single nuclear RNA-seq dataset will be available or for public download and analysis?
So the answer is now. SCP-498 at the Broad Single Cell Portal. Alternatively, just type in the search bar heart. I think you'll see transcriptional and cellular diversity of the heart. That's not the raw sequencing data. That's our post-QC data. For most people that want to run through the pipelines that people use on the Syrah or ScanPy tutorials, use those data, please. There's a lot of cleaning that went into these, and that's not for the faint of heart.
I don't have anything to do with Broad Institute, but we are very impressed with your single cell portal. Thank you very much.
They do a wonderful job with it. Their entire team dedicated to making that useful to people. The other thing I will say that, it's a dynamic interface that people can use. So go into that single cell portal. It'll pull up a UMAP plot for you, type in your favorite gene, and it'll show you what cell types it's expressed in.
Yeah, it's so rich, amazing. So Anand's making another point here. I'd argue that you'd want to use a L2-fold threshold, log 2-fold threshold of at least 1.3, ideally 2 or greater. Worth mentioning that atrial cardiomyocytes are a diverse bunch, though they form a tight cluster, and MPPA is definitely one of those marker genes for this cluster.
Agree, Anand.
Yeah, I think Anand was answering most of your questions here, I should say.
What are you doing to me, Anand?
Very keen.
Okay, at least it's having one voice. Okay, super interesting the way that you've showed segregation of EQTLs per disease condition. Congratulations, simple and to the point. I would add to that as well, Nathan. I really quite like the way that you demonstrated how this rich data set could be applied, right, to EQTLs, for example.
Sorry.
Go ahead.
Please go ahead.
Oh, I just wanted to add to this point because on your graph, it stood out when you did the EQTL for atrial fibrillation that the higher bar was actually cardiomyocytes.
So I'll speak a little to that. So one thing I want to clarify is those aren't EQTLs necessarily. So those are just a specific gene expression that happens to be nearby to a locus. So we did also look at things that were called as EQTLs from bulk data and identify which cell those arose from, but those things are a little independent. So you can imagine that LD score regression, like I said, is more of here's the rule for what cell types might drive heritability of that trait. So if you were going to look at what cell type to go after to study atrial fibrillation, cardiomyocytes, which I think a lot of people are probably thankful on this call, actually do drive a significant proportion of the heritability for that trait.
Yeah, because it's quite easy to make the mistake to think that it's only atrial cells that are of value for atrial fibrillation, but cardiomyocytes in both LA and LV.
I mean, I will say that we combine the cardiomyocytes at that stage. So we didn't do the breakdown, and part of the reason is that when you break them between the two chambers, the number of specific genes to that chamber, you end up killing your number of specific genes in that case. So we did a combined approach there. I suspect if you strip the LV cardiomyocytes out of there and just looked at left atrial, that plot, it might be a little higher bar on there actually.
Okay. Okay, next question. Would you elaborate how the disease relevance was analyzed? I know you combined GWAS and single nuclear RNA sequencing.
Yeah, it's a tough approach. So basically it's you find a GWAS locus, you look within a given bin window, I believe it was 500 KB, don't quote me, it's in the methods somewhere, and then you look for genes that were within that window that were specifically expressed in a cell type. And basically what you want to do is look for enrichment of cell type specific expression nearby to GWAS loci for a given trait or disease. It's really a global enrichment approach. Like I said, I really encourage people to look at your specific locus of interest also. Those are the rules. The ones that violate those rules are the interesting ones, at least to me.
When the cell type that you least expect comes up, it's something to go back and check on that.
Yeah, those are the ones I like to work on.
Yeah. So Anand's again posting the link for the portal at Broad. That's really useful. Thank you very much.
Thanks, Anand.
Caitlin Hall asks, do you make any considerations towards cells that may be bi or polynucleated? That's a really good question, Nathan.
That's a good question, and I get that one a lot. And unfortunately, we're blind to this. We don't know where a nucleus came from. Not only if it came from a mono or bi or polynucleated cell, but even what part of the myocardial layer a myocyte came from. So I think when I put the different approaches down here, the one that can start to get towards some of these questions are the spatial approaches. So there's commercial solutions. I hope no one on 10X is on here. There's Visium. I'm not super impressed with Visium right now. But SlideSeq V2 has been really outstanding. I think the resolution that we're starting to get towards being able to answer that approach. I'd also say too, if I saw distinct subclusters or gradations within cardiomyocytes, and you go into a subcluster analysis of that, maybe they give you hints of that. And the answer is I don't know, but it's possible to try to look to see if there's anything there.
Well, I'm pleased to say Caitlin's my PhD student, so this is the first question she's asked in the webinar series. So well done, Caitlin.
Good question, Caitlin.
What a boss to point that out now publicly. Okay, Jillian Gray is asking, how about endothelial cell ACE2 in expression? This is referring to your second part of your presentation, which is really cool.
Yeah, so I believe it's minimal. Let me actually go back to the slide to make sure I'm not. Yeah, so our endothelial cell populations actually exhibit minimal expression of ACE2, especially in comparison to parasites. Parasites are far and above the strongest expressors.
It's probably not saying that it isn't expressing it though, right?
It's not actively expressing it. I mean, the one thing you need to say with all of this, we're not measuring protein level. So things that are stable that sit on the membrane, we can't account for. But at least as far as active expression, and again, it's relative, it's not absolute, that parasites make way more of this than endothelial cells do.
Yeah, right. So actually, to be fair, I've seen some data that also consistent with that. Okay, so next one is, do we need a special permission to register and be able to download data from Broad Institute? Or just
so Anand answers that right afterwards. So thanks, Anand.
Oh, yes.
No, it's you register with any Gmail account and you'll have access to those data.
Okay. Well, only Gmail account, why?
Yeah, they use it for authentication, I believe.
Okay.
And then he answered Caitlin's question too. I'm gonna give you a call after this and that.
Okay.
I think,
Sorry, Neville.
I think he's hijacking your presentation.
So anybody who doesn't know, Anand is an outstanding expert in kind of models of ES-derived cardiomyocytes and titin biology in particular. So if anybody is interested in screening things against on these high throughput platforms, Anand, in addition to single cell, is also an expert in that.
Oh, fantastic.
You got a free plug out of answering all these questions.
So next one, excellent data. How do you account for nuclei multiples in your analysis?
So in this one, basically it'd be removed at early QC stage. Newer analyses use a tool called Scrublet that attempts to identify cell types that exhibit signatures reminiscent of two or more cells that are actually smashed together. So in this case, it's a little more crude, but you'll notice in the future iterations that at least visually, the distance between two given clusters will be greater. And that tool has been incredibly helpful. So we give a free plug to Scrublet is the name of the tool.
Okay, next one is thank you. Nice talk, Nathan. What is a general consideration on threshold for mitochondrial gene, for example, percentage UMI, particularly when heterogeneous populations are involved? We see some highly abundant genes tend to show beyond its main clusters. Do you see this? How do you deal with this?
Okay, I'm a little confused about the second half of the question. So if you want to follow up with that one, I can. But at least the first part I can answer. Conceptually, the mitochondrial gene contamination I think was initially proposed for single cell experiments. Given that if you see a lot of mitochondrial content, then maybe you're seeing an apoptotic cell or the like. I will say for us, the mitochondrial gene count, I think we cut it off at 5% for this one. Again, don't quote me on that, it's in the methods. But I think a lot of our other QC metrics, mitochondrial gene count kind of went with abnormal number of exonic reads or our other kind of QC metrics that we use to call cells out. So oftentimes those things converged on a decision that we were not going to use that cell.
Okay, we'll go down then to Matt. Matt works with me. So Matt's asking, Matt says, great talk. Thank you. Important advance in the field and very concisely presented data. Kudos. Now a great number of single cell cardiac studies, initially in mouse, more recently in human. I wonder how you feel your data fits in with the data in recent studies. Are we at a stage where some kind of cardiac single cell meta-analysis might be possible? He's throwing you a challenge.
Well, I don't want to. Here's what I'll say to you, Matt, is that we did a single platform from samples that we processed ourselves in an identical fashion, and that it was extraordinarily complicated to get them to go into anything that resembles that map that you see in the paper. And the thought of adding in data sets generated by others is a challenge that I honestly terrifies me a little bit. I think it's possible, but it requires a dedicated effort to do some kind of meta-analysis like this. I mean, also the technologies are not static. So this is V2 of 10x. Other people are doing SeqWell or SplitSeq or 10x V3 or other various approaches. Each one has its own nuances, and the thought of meta-analyzing those data, at least through a common analysis pipeline, would be an incredible challenge to me. Now, then, can the conclusions be compared? Certainly, and I think if you see more concordance between studies, that would lend confidence when you want to study your gene in a given cell type.
Yeah, I mean, to that point, Nathan, I do kind of wonder, are we going to see more and more single cell? Well, we are going to see more and more single cells, right? But what more and more new things are we going to learn at the end of the day? Where do you think all of this is going to help?
Yeah, so I mean, we're getting close, at least from this methodology, to understanding what the nuclear transcriptomes look like. And I think, ours and the effort that's obviously somewhere in review from the HCA folks, led by Sarah Teichman, is, that they'll meet some kind of concordance of that's what the data are going to be. And then, where it goes from there, I mean, obviously we're working on disease phenotypes in humans. You've done some work on disease phenotypes in humans as well. From a model system standpoint, we're starting to find some interesting uses of this for really nice experimental models in mice to say, how can we use this technology to analyze things that we never could before? The new frontier in this, which is a nascent thing as well, would be, I think, the spatial stuff, especially for unique regions of anatomy within the heart or within other tissues, that combining this blind approach with spatial reconstruction of where those cells sit in anatomy is really the thing I think we're going to see over the next couple of years out of this style study.
Nathan, the spatial one that we have come across at our Genome Institute is the one where you can look at it on even a single histological section of the heart tissue. Is that the one you're referring to, the sort of technique?
Yeah, so there used to be one that was spatial transcriptomics, where they're basically spotting probes and then you can reconstruct thereafter, but they're not single cell, and no spatial thing is truly single cell at this point. So what you capture are large regional differences in the transcriptome. So spatial transcriptomics was purchased by 10x Genomics, and they've improved that a little bit, but still it's not single cell. And there's diffusion and things that happen with RNA from these approaches anyway. Obviously, the only true intact in situ approach is to do something like RNA scope or the like. But SlideSeq V2, I believe, so it's developed by Fei Chen and Evan McCosco, who used to be downstairs of me at the Broad. Now they're a little more distant from me. I think the resolution of their approaches is much, much better. Look up their preprint, just came out. I'm really excited to try to implement that for our spatial stuff.
Sounds very techy. Nate, the other possibility is looking for rare cell types in the heart. Do you think that, that this is a good approach with single cell going down to try and find some of these things?
I think so. The one problem with that maps like this actually help with is to find rare things, you need to get rid of the things that are very abundant. So doing negative selection of cardiomyocytes, I mean, you've used PCM1 as a marker for cardiomyocytes. Oftentimes people use it for enrichment. I would love to use it for depletion. I've seen enough of cardiomyocytes in my single cell RNA sequencing data at this point. So as you can start to remove these more abundant things, we can get at things, immune cell diversity, get it better of the cell subtype diversity within endothelial cells and the like. That gives us an approach to look at these more rare cell types. If you were going to do it like this, just such a large percentage, 70% is dedicated to cardiomyocytes and fibroblasts, which also happen to be more transcriptionally complex. And just from a cost standpoint, you have to find a way to get rid of them, unless whatever group has an inexhaustible budget, then feel free. But,
it'd still be a waste of.
I think so. Yeah, it would be an analytic headache for whatever your poor analyst is doing to deal with all the cardiomyocytes. So,
I mean, that's a fantastic approach that you've thought about from the depletion angle.
Thanks.
So I think this could be the last question, right, Davor, from Matt up here.
From Matt, yes. But there's more questions that just come in.
Sorry, I missed a few more. So,
No.
So Matt's asking here also, could you give a little more information on why one needs to be cautious about subclustering? Or even are there cases when subclustering could be appropriate?
Subclustering is always appropriate. But the application of those subclusters to understanding biology, you need to be very cautious. So, I unfortunately don't know what part of my talk, faded in and out. The resolution of clustering is merely changing a single number in the clustering algorithm. And essentially, I can make every single cell be its own cluster. And biologically, that's not the case. So I think it requires, do some fine tuning on your cell subtype of interest. And then importantly, when you have something that you think, replicates across multiple individuals, seems to hold up to that kind of sensitivity analysis, that go back and validate this thing to make sure it's not an artifact. Before, dedicating a year or years chasing down something that's an artifact, spend the month making sure that that subcluster is real.
Very sound advice. So Yichang's Chang is saying, thank you, Nathan, for the comments on mitochondrial gene levels. Other question is, cells with high abundant genes, for example, MYH6 in cardiomyocytes, may have some cells or nuclei showing up in myocyte clusters, although in very low frequency. Did you see that in your study?
So you're saying that genes that have a lot, or cells that have a lot of a given gene might be classified in a different cluster? And yes, that happens, especially with a large number of cells like this. There's two possibilities. Number one, kind of clustering algorithms and dimensionality reductions aren't perfect and they might misassign a cell. The other case is that contamination might be, that expression, even if it's high, high expression in a given cell might be 5 counts. And just from a probability standpoint, that's going to happen sometimes. So an endothelial cell might have significant contamination in a given gene. So that happens in the background. The one thing I'll say to you about that, though, is anytime that occurs, it's going to bias you towards the null. It's going to pull all these things together. So it's rare that those type of spurious findings would drive things different in a way that wouldn't be true. So.
Okay, another one. Excellent work. Thank you. Curious to know if you're able to see enrichment of a set of genes or pathways in COVID-19 patients.
Okay, so we're not actively currently looking at postmortem or biopsy samples from any COVID-19 patients. I'm aware of a lot of groups that are working on this as some of the tissues become available. But I will say generally, gene sets and pathway analyses have been applied to these and other type of data. I expect we'll see a lot of this stuff in the next year.
The data that you showed us at the end of your talk was unpublished work. Is that right, Nathan? So
it's currently up on medRxiv. If anybody would like to have a look at that, it's under review currently. I'm hopeful it'll be available in the same way that this is, that this one is too. The one caveat to that is that unfortunately, we looked at those analyses in isolation. So the global analyses of those disease subtypes will be coming at a later date. So we'll only be able to release the data for the genes within that publication. And that's part of, our partnership with Bayer they have their window to examine those data as well before publication.
So Nathan, sorry, if you don't mind, what was the kind of disease status of these patients? You said that these were not patients that have passed away. I mean, these were patients that presumably survived, and to what extent did they respond to COVID? Were they just
okay? So I think
symptomatic or
yes. So let me clarify that. So those were non-COVID infected patients. Those were people. The DCM or HCM are people who went to transplant.
Oh, I see. Sorry. Yes.
Yeah.
Okay.
And within HCMs, you'll see individuals both with true failing and still in hypertrophy. So kind of you get the both of those. So when you're looking at the data. The COVID stuff, most of the samples that I'm seeing from the various groups will end up being postmortem samples from people. So we'll see what happens with those analyses going forward.
And very little effect that you've seen in terms of ACE2 expression, in patients that have taken ACE receptor blockers. I mean, that probably fits quite well with other people's data. I've sort of seen similar findings, or was that your findings that I was seeing?
Yeah, they might have. It might have been ours. I think some other groups have investigated this as well. I believe I want to say it was the Dimmler group that just came out with a similar study.
I read this information somewhere, but I can't remember where. So that's encouraging.
Yeah. So there's one European Heart Journal. I think just put out a paper similar that found that our case were ACE inhibitors rather than receptor blockers. And again, within the context of hypertrophic cardiomyopathy, so we haven't run enough non-failings that are to deal with the pharmacology. I think it's another interesting point going forward to examine what, long-term ACE inhibitor pharmacology does to expression profiles in the heart is another interesting project outside of this given thing.
Thank you.
And Nathan, are you saying that you're doing single cell sequencing on the COVID patient samples? Is that what I hear you said?
I am not currently. I know that there are some biobanking efforts going on here in the States from postmortem. I would expect that some of these approaches would be taken. I'm curious to see what it looks like. I mean, there's a lot of debate for, the cytokine storm versus primary myocarditis, hypotheses right now, and we'll see if some of these approaches can answer some of that.
Yeah. Okay, another question here. RNA transcript, shortest length of transcript detected.
Yeah, so it looks like that pairs with the next question as well. So generally the prep for next gen sequencing libraries, you're going to be limited to things that are above 400 or so nucleotides is about what you're going to be at. So unfortunately, most of your microRNAs are not going to be picked up by this. You'll have to use a different approach.
Okay. I think this then would be the last question.
Yes.
Daryl is saying, excellent presentation as always. Once you account for batch effects, what clustering algorithm was used to define number of cell types? K-means clustering or something else?
Yeah, so I think we use Louvain clustering here. So you basically, in that case, you're setting it's a unsupervised machine learning clustering approach. K-means you set a fixed number of clusters and then it assigns them. This one we kind of let go based upon a resolution that you assign. The newest methodologies use Leiden clustering, which is another variation of or alternative approach to Louvain clustering. So we don't set a number of clusters. We let it tell us how many clusters there were based upon a resolution. The K-means will be an option in most of the outputs that you can set a fixed number if you'd like.
Okay, I think that is the end of the Q&A. It's been very long, and thank you so much.
Sorry for staying up so late, Roger. We appreciate it.
No, not at all. Don't say that. But it has been really enjoyable. This is something that I have been looking forward to all day, in fact, and rereading your paper as well before I come on, just so I know what questions to be asking. Thank you so much, Nathan, for doing this and taking us through. Thank you, Davor, for as always hosting us through these sessions here. Davor, you should have the last word.
Well, I mean, I would like to first of all apologize that obviously we did have sound issues. It looks like it was related to sharing the presentation, because as soon as you stop sharing, we can hear you really well. But I also saw from some of the comments that people, for example, in Georgia could hear you very well. So it could have been a regional effect as well. Who knows? So I apologize for that. Let's see what the recording is going to be like. We may have to invite you to do another one, if the recording is not great. But I really enjoyed it, and we had so many questions, even with the sound issues. I don't even want to think about what time we'll be here till, if actually there were no sound issues, because clearly it's an exciting topic and you're a leader of your field. So thank you, Nathan. Thank you, Roger. Without you, we wouldn't have these interesting discussions because I wouldn't know what to ask. so thank you both, and really appreciate your help. And thank you to the viewers as well.
Thanks, everyone.
And see you all on Monday.
Bye, guys.
Thanks, everyone. Bye.